All right, guys, so in this video, I'm going to be going over the last two FRQs on your final ones for this week. And for these ones, it's not too terrible. They're both pretty short, but it's kind of a good overview of fermentation. So this one reads, this one that I'm going to be going over first here is actually going to be number 7. So this one mentions that a couple of different types of bacteria. So it says many species of bacteria grow in the mouth of animals and can form biofilms. Within plaque, the outer layers contain high levels of oxygen, and the layers closest to the tooth contain low levels of oxygen. The surface of the tooth is covered in a hard layer of enamel, which can be dissolved underneath acidic conditions. When the enamel breaks down, the bacteria in the plaque can extract nutrients from both the tooth and cause cavities. So this tells us that we have two types of bacteria. The Streptococcus mutans thrive in the most innermost anaerobic layers of the plaque and are associated with cavities. Other types of bacteria, the Streptococcus sanguinis, compete with the S mutants but are unable to thrive in acidic conditions. All right, so here we go. We got two types of bacteria. So the Streptococcus mutants thrive in the innermost anaerobic layer. So that's a key word you want to know. Anaerobic means that there's no O2 available. And those are the ones that are going to cause cavities. The other type of bacteria, the sanguinis, compete with the mutants in terms of they compete for nutrients, but they cannot thrive in the acidic environments. All right, so let's look at this first part. A says identify the biochemical pathway that the mutants, so again, the mutants use for metabolizing sugar. So metabolizing just means to break down. So you might want to remember what that word means. So we're going to be metabolizing sugar and describe how that pathway contributes to the low pH in the inner layers of plaque. Okay, so no oxygen. So the only way that, well, a couple of things here. Prokaryotes, they don't do respiration. They can only do glycolysis, and then how they process that sugar is going to vary from bacteria to bacteria. So just want to remind you, glycolysis is there's no mitochondria involved. Remember right here on the right, we have prokaryotes. Again, these bacteria cells are prokaryotic. There is no nucleus involved. Their only source of making any ATP is from glycolysis. So in terms of this, lactic acid fermentation, you're taking glucose, they make pyruvic acid. And the point of glycolysis, remember, is that you have to regenerate NAD+ to even do glycolysis. But one of the byproducts in this is lactic acid. So let's answer this first part really quickly. What is the pathway that it uses? So the first thing that you would state to get a point, you would say that they do glycolysis. And to even further make sure you get the point, I would even mention fermentation because there's no oxygen available. That's really what it wants you to at least be able to identify. Both would earn the point, but describe how the pathway contributes to the low pH in the inner membranes of the pathway. Well, you're actually increasing lactic acid, because one of the byproducts in this would be the lactic acid byproduct, which would lower the pH. So those were how you get those first 2 points. Now for B, it says normal toothbrushing effectively removes much of the plaque in the flat surface of the teeth, but cannot reach the surfaces between the teeth. Many commercial toothpastes contain alkaline components, which essentially raise the pH of the mouth. Predict how the population size of the S mutans and the S sanguinis in the bacterial community in the plaque between each teeth are likely to change when these toothpastes are used. Okay, so a couple of things about this one. You got to go back and see really what this passage tells you. So the key thing on this is that it would actually lower the concentration of the mutants because you're actually increasing the pH. And this tells you that they thrive in those acidic environments. They have a byproduct of lactic acid. And so that population of bacteria would actually decline. But it also, remember, it asks you about the sanguinis population. And it tells you that the Streptococcus sanguinis, they compete with the mutants, but they're unable to thrive in the acidic environments. So basically, the more of the Streptococcus mutants you have, the less of the sanguinis you have, because it tells you that they compete with one another. Well, all of a sudden now you've taken out the mutants because you've increased the pH. So to earn the second point here, you had to say that you're going to be increasing the concentration of the sanguinis. And this is because they can, you can infer that they do thrive in the higher pH environment. They don't survive in the low pH. So that's how you earn those 4 points. So hopefully that one wasn't too terrible. Let's knock out this last one. This one is about analyzing the graphs, so let me make sure you feel okay on this. So this one says microcyst aeruginosis is a freshwater photosynthetic cyanobacterium. When temperatures increase and nutrients are readily available in the pond habitat, the bacteria undergo rapid cell division and forms an extremely large visible mass of cells called an algae bloom. The cells have a short lifespan and is decomposed by aerobic bacteria and fungi. Identify the metabolic pathway and the organism that is primarily responsible for the change in oxygen level in the pond between times 1 and 2 and between times 3 and 4. So we're looking at oxygen levels between 1 and 2. So what's going on here? We'll answer that first. Then we got to figure out what's going on here between times, or excuse me, 3 and 4. So we got to figure this one out. So we're looking at our oxygen levels. So O2 levels are represented by the solid line, and according to this one, it looks like the O2 levels are going up. And we got to figure out what cell is going to be responsible for that. So cyanobacterium, if you forgot, they're a type of bacteria that can do photosynthesis. So just kind of want to remind you guys about this. Photosynthesis, remember the byproducts, you create really two things. You create oxygen as a byproduct, and you create glucose. And in terms of respiration, you're going to be consuming that O2, so O2 levels will go down, CO2 levels will go up. So in this case, to get that first 2 points, we got to figure out what's going on between 1 and 2. So we're identifying the metabolic pathway that this is doing. So for this one, I'll answer here for the first part. Between times 1 and 2, the pathway, what's causing that increase is going to be photosynthesis, because it tells you it's cyanobacteria, so they're capable of doing it. So identify and what species is responsible for that. It is going to be the cyanobacteria. Because if you look to the cyanobacteria population is increasing, so as that concentration is increasing, the oxygen levels are increasing because you're actually increasing your oxygen levels, okay? Now, let's see what's going on between times 3 and 4. Between 3 and 4, it's pretty evident now that your O2 levels are declining. And as well as this, your decomposer levels skyrocketed kind of in between here, because there was essentially, as your oxygen levels started to decline, slowly thereafter, all the cyanobacteria was declining. So maybe there's a lack of sunlight available or something causing photosynthesis to decline. So what's the metabolic pathway going on between 3 and 4? That would be cell respiration. And what is responsible for that? That would be your decomposers, because your O2 levels are going down. Cell respiration uses O2. So pretty simple FRQ on that one, but just kind of good representation of how to analyze and read your graph. A lot easier than you probably would have thought. See you guys in the next one.